The overall goal of the following experiment is to use a combined proteomic approach to identify potential prognostic markers and/or therapeutic targets in human diseases. This is achieved by isolating primary leukemic cells from patients' peripheral blood carrying good or bad prognosis and expressing both CD19 and CD5 surface markers to use for proteomic analysis. As a second step, leukemic cell lysates are resolved by two-dimensional electrophoresis, or 2DE, and the comparison of proteomic maps allows the identification of spots differentially expressed among good versus bad prognosis patients, which can subsequently be identified by mass spectrometry or MS. Next, different assays can be used in order to validate and dissect the role of the identified proteins in the natural history of the disease. The results show that different phosphorylation status of HS1 detected by 2DE-Gels correlates with the clinical outcome of the patients.
The implication of this technique extends toward the therapy of chronic lymphocytic leukemia, because it can help to identify protein involved in the progression of the disease that can be exploited as prognostic marker and/or therapeutic targets. Demonstrating this procedure will be together with me, Pamela Ranghetti and Federica Barbaglio, two technicians in the lab, and Umberto Restuccia, a postdoc from an external lab.
Prior to starting this procedure, collect peripheral blood in vacuum.